beautiful day, absolutely beautiful. Anyway, I'd like to say one thing here, that is that I have graded the exams and the grades have been posted onto Sakai. I was really impressed actually with the scores. I mean, really impressed. I think that maybe I didn't make a test that was challenging enough for everybody. You really showed me a thing or two. And so, I mean, it was really, I mean, there was, yeah, I think the average was was a B, and typically in this course, the average is a C. And so that's, or C plus. But actually, that's extraordinary, though. So well done to you. I think that shows some excellent scholarship. And so, if you still feel badly about your grade, though, don't worry. There are still opportunities in this course to still pick up some points. For instance, we have the final, which is coming up. The labs haven't been tallied into your final score yet, but the labs and the final project. I mean, the final project. There's no final exam, but the final project. That is yet to come. I'm still deciding on what to do about the second exam that we have here. It'll probably be something that's similar to the first exam. I'm thinking that will come up in the next couple of weeks because we don't have that much time here. And so that will be that. But still, there's that score. But what I'm trying to say, though, is that there are still points that are coming. And so if you feel disappointed in the score that you got from your exam one, then don't sweat it because you still have opportunities to still get some of your points. I mean, it's not the end of the world. So, but anyway, but still, if you'd like to discuss your exam score with me, then please let me know, and I'd be happy to do so. I would like to spend some time, though, actually, to talk about one problem. We can look at the other problems too, but this is the exam that you just that you took. So, one, and the question I wanted to talk about in the beginning was, or the first question here, where I'll just read it to you. It says a researcher extracts a random sample of DNA from two different organisms. The extracted samples are about 10 bases long each. Amazingly, there's a subsequence of 5 bases occurring in the same order that has been discovered in both of these sequences. Now the question says, is it significant to find this common region of 5 bases? Why or why not? And in your answer, please discuss what can be learned about the relatedness of these two organisms by the discovery of this common region. Okay, so this is actually a very interesting question. And the reason why this is interesting is because if you were looking at this strictly by numbers, as a statistician, if you're driven only by numbers, then to have half of the sequence, 5 bases out of the 10, the same between these two samples would be significant. Half of it's the same. There must be some reason for half of it. But if you think about it, again, as a statistician, 5 bases actually isn't much. Those 5 bases over the evolutionary period of that sequence, that's 5 mutations that just happen to be at that same place. For whatever reason, they stuck around, and so maybe those mutations are what makes the sequences the same. So it's random. It's not enough information. But most importantly, though, let's go back to this thing here. It's a random sample of DNA, and so we don't even know if these are the same pieces of, or in the same areas of the genome for these or these sequences are coming from the same genome. So in other words, it's like they're just pieces that I'm pulling out of a hat, basically, like pieces of paper pulling out of a hat, and saying, oh, I happen to find 2 pieces which are exactly the same. I have no idea where they came from. I don't know whether they're from the same gene. I don't know anything about them. The only thing I do know is that they're 10 bases long, and half of those bases are the same. And so, is that a significant find? Well, I would argue that no, it is not a significant find, simply because it's not long enough. The 5 common bases is not long enough. But even then, 10 bases is still not long enough. Think back to the BLAST E-value score, where the value of E will go down to values that are close to 0 when the sequences are long enough and similar enough. And so it's really a measurement of not just similarity, but saying, yes, they're similar, but it wasn't due to chance. And so I'm saying that in this particular, your E-value would be quite high if this were a BLAST, because it could be by chance that you get 5 bases which are common to both sequences. And so I think I just took off like maybe 3 points off the question. If you had the right kind of argument, then you still got partial credit, but otherwise, if you said it wasn't significant, then you lost 3 points. Or if you said it was significant, then you lost 3 points. But I would say that it's not significant. However, if you find like a random piece of DNA that comes from, or let's say you randomly choose a piece of DNA in 2 different organisms that come from the same location in those genomes, or the same gene, for instance, and you happen to find that the genes that you choose randomly, let's say you take gene A from organism 1 and gene A from organism 2, and you find out that those 2 genes are exactly the same. I mean, you know that they're the same, but you find out that the actual composition of those genes is the same, then that would be significant. And also, for instance, you would imagine that the gene would be actually more than just 10 bases. And so you have to think of this in terms of not just what the numbers say. And the question is actually kind of it was kind of a trick question, I guess, in some ways. But a statistician, though, or a bioinformatician is not necessarily just looking at the numbers, but a bioinformatician is also thinking about what's the most logical explanation behind this sequence? How could these sequences actually be the same? And so what I'm asking you to do is to become something of an expert on how sequences could possibly be different, and then use that knowledge to infer some kind of rationale or some reasoning behind why it is that you found 2 sequences which are the same. Is it something which is noteworthy, or is it just something that perhaps you just found? I'll leave it there, but if you have any questions over this, please let me know, and I'd be glad to discuss it with you, or we can come back to this question in class if there are enough questions. Another question I think that everyone nailed here, which was actually quite good. I won't go through all of these questions, but the differences between the global and local alignments. I think everyone had it basically correct. But the global alignments is you're looking at sequence by sequence, whereas local alignments you're looking at in the sequences what parts are the same. So the sequences themselves, the whole sequence might be different, but you might find kind of subsequences inside each of these big sequences, and you're able to find those subsequences which are the same across multiple sequences. And so that's really the big difference. Now, this question, actually, I asked this question in another bioinformatics class, and it seems like a number of people missed it. And so I'm so happy that basically I'd say that an overwhelming majority of everyone got this question correct, and they got all 10 points. Good morning. So that makes me very happy. So anyway, if you have any questions of your test, let me know. There are, I think that when you look at this question, or when you look at your test in Sakai, I think it's there, and maybe it's not there. Maybe I have to let it. Tell you what, I'll look into that. But I think if it's not there now, I'll make it available so that you can see your old test, and you'll be able to see some of the model short answers there, and that will help your thinking. And we're going to kind of continue the conversation that we had on Monday, which was a discussion about gene prediction. This is actually a very exciting area of bioinformatics because you're really looking at, it's like you're a detective here. It's like you're Sherlock Holmes, and you're trying to find out, who done it, who made the protein, who done it, who made that protein. Was it you? Was it you? Was it you? And even then, but in this case, it's even more tricky in that we don't have a protein, so it's more like there's no evidence, or there's no evidence of a gene existing, but yet we want to find out whether there's any chunks of code which could be genes. And so that's why we call this gene prediction here. So we're going to continue going on here. And we kind of brushed on some of the ideas of like alignment base, where you're looking at, we've discussed this without even knowing it. The global and local alignment, for instance, we can predict genes based upon the idea that if you find a region of code in, say, organism A, which is also found in organisms B, C, D, and E, if you find that same region, and you know that region happens to be a gene, then you can infer with some educated guess here, with some education, that subsequence is a gene. And so I never saw the gene being used in organisms, but I recognized the sequence. And so since I knew that sequence was there in other organisms, I knew that, for instance, that was a gene. This is actually kind of similar to if you go to like a nightclub here, and the DJ is playing a song, and you hear some other song mixed into the music that you're dancing to. You might recognize that as a song that you've heard somewhere else. And you might also recognize, like, wait a minute, this isn't a dance song, but they've done something so they kind of speed things up here. And so it's like you recognize the song, but it's not the same song that you knew before. It's really, it's like you're recognizing something of the melody or whatever it may be, the beat or the voice or something. But you recognize that it's something you've heard someplace else before. And that's really how you're finding genes in many cases when you're using this alignment base. You don't recognize exactly the same sequence of the gene, but you do recognize that you've seen it before. I've seen something like this before, and then that means you have to do some kind of research to see whether whatever you thought you saw before, is that really similar enough to the stuff that you see now to say, yes, this is a gene. And so that's why you use this alignment-based system here. And again, we use our friendly system BLAST. Then we have, for instance, let's see what else. So we have the sequence-based, and the sequence-based system is very, very similar, where you're using what the ORF, or the Open Reading Finder, the Open Reading Frame Finder, and that is looking at landmarks, like specific types of landmarks that exist in code, and saying, I recognize this landmark. When I see this, I know that there's a gene that's going to come up. When I see this, like the Shine-Delgarno sequence, for instance, when I see that, I know that something's going to happen. I know that I should expect some kind of gene starting. And it actually makes sense to find these things, because if you are looking for, if you're using these landmarks, imagine how does the biological machinery that actually unravels the DNA at exactly that point, does its transcription to make the RNA. Then after that, you have this RNA sequence, which is then created into or then transcribed or translated into protein. It makes sense, like, how does the machinery that does that work know where the genes are? So there must be something physical that's there, like some signal, as they call it, some reason to believe that this chunk of code is actually a gene. There must be some signal there, like a common sequence or something that I guess they call it the RNA polymerase would bind to. Something that some molecule would bind to the DNA or the RNA. There must be something there to suggest that this is the spot where the binding would happen. And so that's this sequence base, where you're looking at specific types of beginnings and ends of what you'd expect to find for genes. And so then, in this case, we're looking for start and stop codons. Now, one thing I'll say about start and stop codons is that it begins with a start. And yes, every gene has an end. It has to, otherwise it would just continually it would continually create protein without any stopping. And if you didn't have that start, then the protein may never get initiated. However, you can have other types of start and stop places in sequences which appear to be genes, but they're actually not genes anymore. Maybe they were once in ancient times. They were genes, but they're no longer genes. They don't do anything anymore. They've been somehow disabled, yet they still have the start and stop codons in place. And so that means it's like a computer program, I guess, that has the syntax, but it has the syntax in place. The variables are all being defined, yet something never runs this function. It's like it's just sitting in your code and it never gets run. And so yes, you can find that gene, but it doesn't mean anything. Excuse me. And then the next thing we have is the probabilistic approach to finding genes, or predicting genes here, which is really using kind of a combination of stuff. It's this word here, combination, but you're really using like different kinds of information. This is probably, I would reckon, probably more reasonable, a more reasonable approach to predicting genes, and that is that you're not using just one thing to find that gene, but rather you're using a host of different types of evidence. And then when you find that evidence, like I'm finding this Shine-Delgarno, I'm finding some other stuff, I'm finding the stop and start codons, I'm finding some other pieces of landmarks that I recognize. And then you ask yourself, well, what are the chances that these things would be there if it were not a gene? And so you're kind of like, you're thinking in terms of, well, could it be that this stuff could just show up randomly in the sequence? It could be that, yes. It could be that stuff like that sequences like the Shine-Delgarno, I mean, it's not very long, it's only about 6 bases, I guess, wasn't it? 6 to 8 bases. But it's like it could just show up randomly. But could it show up randomly with some other stuff, like some other landmarks that you'd expect to find with a gene? maybe not. So that probabilistic approach you're using is kind of describing that the gene is showing up. What we're talking about today, though, is the content-based. So this is actually kind of more detective work here, and perhaps more into the unknown, but you're looking for patterns and frequencies of stuff. You're looking for, like, for instance, what are the imprints, I guess, of a gene? Like, if you have a gene that shows up, are there some kinds of, I don't recognize the gene itself, but is there something that I should be looking for to see that a pattern is going to begin? So, for instance, getting back to the nightclub idea, what is a pattern that you would expect to see as you're walking toward? Let's say you're going out in the town with your friends. You're going to a nightclub. You're about ready to start, going inside and dancing the night away. As you're getting closer, what kind of patterns would you expect to see? What kind of trends would you expect to see to suggest that you're actually getting closer to that nightclub? Well, for one reason, or for one thing, you'd expect to hear music, probably loud music. You'd expect to hear a lot of kind of dance music. Another thing, you might expect to see people dancing, and for instance, you might expect to see special lighting. And so what I'm saying is that there are certain types of things that you would expect to see if you are going inside a nightclub. But if you if you may not recognize this nightclub. Maybe you've never been there before. Maybe you've never been on their dance floor. You have no idea what the place looks like inside. But you can certainly infer that there's probably a disco ball, there's probably a DJ, there's probably a speaker system, and there's probably a lot of lights and flashing and people dancing. And so those are the kinds of things that come together to make a dancing place, like a nightclub. So you're using kind of patterns here. You're saying that this is what I would expect to see in terms of a gene. I'm saying that I should see a gene. If I see a gene, I should see these particular aspects to suggest that the gene is there. And so, for instance, we can go back to some of our structures that we're looking at. If we're looking at, for instance, the genes that exist in prokaryotes, which are typically bacterial types of cells. So, for instance, you have a coding sequence, which is this is the open reading frame, and in this particular coding sequence, that's your gene. That's the part that does something. It's supposed to make protein. But again, you have this ATG, which is your methionine, and you have this TAG, which is your stop codon, your start and stop codon. Your TAG is going to be your stop codon. So remember that most proteins begin with something called methionine, which is your amino acid, which really is kind of it's M for your codon table, but basically it's the beginning of most proteins. Now, this isn't to say that all proteins begin with methionine. It doesn't work that way, but most proteins do begin with methionine, which is actually kind of interesting. But here, though, in the prokaryote cells, you do have this. You do have a Shine-Dalgarno sequence that you can look for, which is this AGG, which is upstream from the. Or is it? Where is it? It's not far from the ATG that you would expect to find your methionine. And so, in other words, you have this to look for. One thing that's kind of interesting here is that I don't know why I'm putting these things here. This is for this line here. You have these highly conserved parts of your landmarks. Like, for instance, here is the Shine-Dalgarno sequence immediately before the start codon. And so again, it's like getting back to the nightclub idea, is that I've never seen this nightclub before, but I do know that I hear music, I see lights, I see a DJ, it must be the dance place. And so here, for instance, every single nightclub will have that, a DJ, lighting, and sound, so that you can dance. And so it seems like every single, or most genes that you'll come across in prokaryotes have this sequence, which is immediately before the start codon. And so therefore, once you have that, then you have an idea that, oh, wait, I'm about to start finding a gene here because I found this landmark. It's got to be around here somewhere. And this is actually kind of interesting here, where, for instance, you have some other types of landmarks that you can look at. These are this is the Pribnow signatures, this is called the Pribnow Box, which is after this discovery in 1975 by someone called David Pribnow. I have to say, on a side note here, when I think about, like, for instance, making a discovery like this in 1975, that was probably without much help with computers. Probably the work was done manually. And so the people who did this work back then they must have been really just dedicated, because this would probably take a long time to actually find something like this. But still, though, this is your Pribnow site, and that is another indication that you're going to start seeing something that codes for protein. And so this is also something you might find in prokaryotes. So that's interesting. I guess you could call this something of a promoter. A promoter sequence is really saying that it's getting the biological machinery ready to start working with either copying or transcribing that DNA, and then making the protein itself once that DNA has become RNA. And so you have this machinery that has to stick somewhere. It has to know what it's doing. Another thing which is kind of interesting about when you're finding gene structures, or when you're looking at gene structures in these eukaryotes here, is that not all these in eukaryotes, so the cells we were looking at before are prokaryotes. These are bacterial cells. It may be more easy to find some of these so-called promoters or initiators of genes, but in eukaryote cells, it's not as easy, because there's a lot more change. So for instance, here you have not all promoter elements are present in all genes. That makes things complicated, because now I can't find anything like the Shine-Delgarno works in prokaryotes, not eukaryotes. So that means the Shine-Delgarno would work in bacterial cells, not in eukaryote cells, which are basically what you and I am made up of, and plants and other things too, but it's a more complicated cell. And so you can have another thing that makes things difficult is you have these promoter elements sequences here. They vary, and so it's like, I know that there's some kind of promoter that exists here, but I'm not exactly sure what it looks like. But I think, though, that what we're going to see now is that as long as you know that there should be something there, then maybe you can start looking for the effects of this thing, and that's really what we're going to be talking about today. Here we are. So there's no Shine-Delgarno-like sequence in some of these cases here, and so that means it makes it much more difficult to find this stuff here. But if you know that something like the Shine-Delgarno is there, something must do that work. If you know that it's there, then you can start looking around for things that may help. There is something called the Tata box, which is something of an initiator of genes. And what that does is it allows the certain types of biological machinery to adhere to the DNA and then to start getting their work done. And that's what all these things do. But you have something there. But this Tata box is TATA, and it's not very much code. And so, for instance, it could be real, or it could be arbitrarily. It could be arbitrary. It could be that you had like 4 different mutations during the history of this sequence. For which you have TA put together. So you could have that. But chances are, though, if you find a TATA, it might recommend that that's a gene coming up. So what I'm trying to say, though, is that in many cases here, you can find the end of the gene, because ends of genes typically are more recognizable. I mean, it's like you're turning off the gene. And so you can find the end of the genes here. But actually identifying the start is much more difficult. And the reason why that frustrates things a bit is because genes have different lengths. And so you don't know whether this gene is 1,000 or 2,000 or 3,000. You have no idea where it began. You just know that it ended here. But the idea is that if you know where the end is, maybe you can work backwards and work upstream and find the beginning. And yeah, so we'll talk about that in a second. So let's talk about actually what the genes are made up of. In a eukaryote cell, like a cell that you might find in a plant or an animal, not a bacteria, but seemingly every other type of cell, you find that there are areas of code that create protein, genes. Now, in your genes, you're going to have 2 different types of regions, and this is baffling. One region actually does something to create the protein, which is the exon, which is the expressed region, and the other one is called the intron, which is not expressed. The names are kind of, they kind of sound the same after a while, intron. But if you remember that exon, expressed region of genes. And so what I'm trying to say, though, is that inside your code of genes, you have regions that actually make protein, and then you have regions that appear to do nothing. And when I say appear to do nothing, these introns, which appear to do nothing, are actually physically removed by special biological editing machinery, so that you only get exon chunks all kind of lined together, and then you can start making your mature RNA out of that. So your pre-RNA is going to be when you have like your RNA sequence that has your exons and introns all included, but then your mature RNA is when you have only the coding parts of the code all together, and the non-coding parts of the code have been removed. It's really quite baffling, and depending on who you ask, it seems like everybody has a different idea about why you'd have why would you expect to find a piece of code in your gene that does nothing. In fact, you remove it. It's like writing a computer program where you have a whole bunch of functions here that you have to manually remove from your code in order to run that program. But then when you write the program, when you save it to GitHub or whatever it is, that program must have these functions that appear to do nothing. So one thing that I can say that maybe tries to satisfy some of your curiosity about why you find stuff in there that doesn't do anything, is that when you wind up genes around chromosomes, or when you store genes in chromosomes, like DNA, as it gets wound up in chromosomes, that's how it's stored. It's bending around other parts of the chromosome, and that bending, I guess, could cause wear and tear on actual coding regions if it's a gene. If the DNA actually is the DNA that you're wrapping around is actually a gene, it stands to reason that if that DNA code is somehow pulled tight over another part of DNA to make this spool, maybe, for instance, you'd wrap up your DNA in a way such that the non-coding regions are the things that actually take the strain, whereas the coding regions are more or less protected around those areas. And so it could be that introns are there to serve the purpose of kind of cushioning, and maybe the expressed part of the genes are there to be kind of embedded inside these introns when everything is wrapped up. But that's just a theory. Nobody really knows. Another thing that's kind of interesting, though, about these things is that between these intron and exon areas of your genes, you have what they call boundaries. I'm talking about the RNA now. So that means that if I have a piece of RNA here, which is what they call a pre-RNA, which is the RNA from DNA, it's the RNA that's taken exactly from DNA as it is. There's been no editing done. I have RNA that contains coding regions and non-coding regions of code. It's everything together, introns and exons all together. And so between the exons and the introns, you have what they call boundaries. And these are kind of like, chunks of code, for instance, that would signify some editing, I want to say editing software, but editing biological machinery. What you're about to see now is no longer an exon, but now we're getting into an intron, and this piece needs to go away. And then once at the end of the intron once you come to the end of the intron, you'll find again some kind of boundary code, some kind of marker, some signal. I'll use the word signal, something to indicate that this is the end of the intron and now the exon is beginning. And this boundary exists throughout the entire gene. So exon, signal, intron, signal, exon, signal, intron, signal, exon. It's like there's something between each of these chunks. I mean, there has to be, though, right? There has to be some signal, something that the biological machinery would use to recognize that, oh wait, this is the exon, or this is the intron. It's more than just a methylene group that's stuck onto the base in some way here, but it's something more significant than that. And so typically we find, for instance, these are a bunch of GCs. Like a bunch of GCs are found between the exons and introns. And these GCs, remember, those bases have 3 bonds rather than 2. But when you find these GCs, you might suggest, or you might think to yourself, I know that I'm in a gene, and I'm coming across this area where suddenly I'm finding this GC-rich boundary, this GC-rich area. Could it be that this is the separation between my exon and intron, or my separation between my intron and exon? The trouble with these boundaries is that there is not much conservation. That means that there's no hard and fast rule that you can use to figure out how one intron are, what the separation is. when I say a hard and fast rule, so what I'm saying is, like, for instance, when you go to a building, you know that there's a rule that there's a door. You find the door, and when you find the door, you find the handle, and then you turn the handle and you pull the door toward yourself, and the door will open up, and then you can enter the building. That is a hard and fast rule about how to enter a building. Find the door, find the doorknob, turn the doorknob, pull the door open, and you're in. So you can kind of create a rule that a machine could follow to open a door. Now, when you are finding the separations between exons and introns, you do not have the same kind of rules that you can just go ahead and hard code into a program, and you can't tell the program what to expect as you're drifting between exons and intron boundaries. So that's what I mean by there's little conservation, and that is that it seems that the separations they vary, and so it's easy to find out or to write a piece of computer code or machine learning type of training system that will help the computer to figure out where the exons end and the introns begin, or where the introns end and the exons begin. One thing I wanted to show you, though, is that you can see this difference. In fact, if I can go back here, what we see here is that we see these boundaries here. But what we're looking at is, for instance, you can use this tool here, which is called WebLogo. WebLogo is a tool that you can use to kind of compare very quickly different types of sequences. And so the next couple of slides actually use results from WebLogo. But basically, what WebLogo does, you can click on this if you want, but maybe I'll save this for now. But what you have, though, is you have it looks like BLAST. You can put in your FASTA sequences, as I've done over here. And in fact, if you wanted to copy and paste your sequences, you could just do so straight out of your slides here. But what it is like you put in some sequences here, and the sequences are AAA. In fact, you have two sequences of AAA, and then you have two sequences of AAT. And you can see right here, I've actually kind of outlined that T are in two sequences which are different from everything else. All these sequences agree that they all begin with two A's, but they end either in A or T. And the tool here, you get this graphic that looks like this, which says that of all the sequences that you entered, they all agree 100%. And this, by the way, is this bits. That's using information theory. Don't worry about that, but that's really saying how much information that there is. But they're saying that all these sequences, by the size of these A's, they all agree that they begin with an A. And they all agree that the middle base is an A. However, the third base, which is right, the third base is an A and a T. And so you can see in these kinds of readouts, if you have like a long sequence when you're comparing sequences of more or less the same length together. In fact, they may even have to be exactly the same. But what I'm saying, though, is that you can say that the big A and the big A at position 1 and 2, that's conserved. That means everything else, everything is the same there. When I say conserved, it's the same throughout. Everybody agrees that it's the same. They all have the same basis. However, at position 3, that is not conserved because everybody has a different. They all have a different base at that position. And so, getting back to the actual, why are we talking about this? Because these boundaries, if you look at code that's for types of genes, or maybe similar genes, you'll find that you'll find maps that look like this, where you have A, where everyone agrees, and that could be the gene itself. But the boundary itself, though, between the exons and introns, or the introns and exons, could alter. It could be different. And so what works for one gene to tell you where the exons and introns are may not work for the other gene, even though you have so much agreement between the sequences, which is frustrating. But just because you have that, it doesn't mean that you can throw that knowledge away. You could use that knowledge to say, well, I know that the boundaries are not conserved, so there's going to be some play around those areas. Maybe that's how I spot boundaries. And so this is kind of what you're doing here. You're looking at some sequences. In fact, these are the same kinds of sequences that we saw just above, where you can see that in these sequences, the numbers are like probabilities, if you will. They're not, but you can treat them as such. But you're saying here that in this boundary, I can see that, for instance, there's in this part of the code, I have an A and C. All my sequences either differ between A and C at that particular point, but then they have A's and G's and T's, and then they differ again by A's and G's. And so you can see that when one base is like on top of the other, like right here, then that means that those sequences are differing. And so perhaps that is your signal. Perhaps that is what you can use to determine that this is actually, it's the boundary. It's the boundary. Another interesting thing that you can look at, though, is that there's something called the energy signals. Believe it or not, there's actually when we talked about the amino acids physio or chemical physio properties, you can actually calculate what's called the energy value for each amino acid. And so there's a paper that I just came across just the other day, which is talking about the energy itself from these amino acids, or in the sequence itself. That energy also fluctuates as you're going from one area to another. So that means that you could use that as another signal. So when I use the word signal, I'm saying that it doesn't come out and just tell me that yes, this is what's going on, but a signal is kind of an indication that something is going on, and that more research could be done to use that signal, to exploit that signal, to determine that yes, there's something happening here. Yes, that is the boundary. So so far we're looking at the agreement between sequences and genes, and the energy that comes out of these different regions. And so, if you watch the literature, you'll find out that seemingly every month there's another paper that comes out talking about how do you determine whether you're in an intron area or you're in an exon area of a gene. How do you do that? What other signals are there? And papers are getting very sophisticated. They're coming up with all kinds of techniques. One paper might be like just basically looking for structures. Another one's looking for energy. Another one's looking for what kinds of bases are there, what kinds of bases are not there. What are the structure? What is the what's the mix of the types of DNA words that you'd find in there? There's a whole bunch of different signals. And so going back to the beginning where we were talking about like the probabilistic method of finding genes here, if I can find this again, we're really kind of still here in some senses. It seems like everything we do is going to be using this probabilistic method where you're kind of thinking, well, what's the chances of this happening, of this signal being shown otherwise, randomly? And so you're looking for patterns, yes, you are, but you're still going back to the probabilities that these patterns are there as a result of some kind of a, in terms of what they have to be there for their, for the boundary. Where are we? I'm getting too carried away here. So yes, and so going back to this here, the boundaries themselves are not conserved. Here you can see this giant GT in the middle here suggests that all the sequence, there's a whole bunch of sequences here. Don't worry about the 1 and 2 bits on the right-hand side. That's just an information theory thing. But you're saying that of all the sequences I'm looking at here, everybody at these middle positions here, they all agree that G and T are at those positions. But then around G and T, you find all this other stuff, which is like I'm looking at the right-hand side of GT, and I see there's an AG, I see there's an ACT, G. I'm thinking these are all the different bases here, and the size of this character is the number of sequences which actually have that base at that position. It's just a visual way of looking at sequences, and so you can see that perhaps this is a boundary. It's not highly conserved, no. But the stuff in the middle is conserved, but the stuff on the outside of GT is not conserved. So maybe all this noise, as I'll call it, is an indication that I'm crossing some kind of a boundary. And you can find that in other cases too. If I can get back to my sequences here. Yeah. So that's the takeaway from that. So you have some differences when you're trying to predict genes between prokaryote and eukaryote cells. Or genes, eukaryote and prokaryote genes. There are some major differences here. You have differences in where the promoters lie. You have, for instance, in the prokaryote gene structure, you have the Shine-Delgarno sequence that marks a start, and that's typically what's there. You have, for instance, you have few interrupted open reading frames. That means it's if you find an interrupted open reading frame, then maybe that's a signal. In the eukaryote cells, for instance, there's no Shine-Delgarno sequence, which is kind of annoying sometimes because Shine-Delgarno sequences are actually quite convenient. But you do have some other types of, you have other kinds of promoters that you can look for here. There's like there are other things you can look for that are hopefully the start sites of your genes, but again, sometimes they're difficult to find because there are different types of promoters. Another thing that's kind of interesting in eukaryotic cells, though, is that nearly all genes contain introns, which is significant because if you're going back to this idea here, where you know that, for instance, there's a bunch of boundaries inside a gene. I know that, for instance, a gene is made up of introns. I know it's made of exons. That means that if I find some region where I'm getting this flip-flop between what I believe is a coding region and a non-coding region in a eukaryote cell, then maybe, for instance, that is a gene. I may not know that if I'm looking at a prokaryote, because a prokaryote is slightly different, but in a eukaryote cell, in a eukaryote gene, I know that those two regions exist in genes. And so if I find that changing nature of the intron and exon in a gene, in a place that I believe is a gene, maybe it is, because I know that genes all have introns. That's not necessarily the case in prokaryote genes because they don't necessarily have these introns. But still, the intron-exon boundaries are still hard to discern because you have to use patterns. You have to do recognition. So what I'm trying to say, though, is that if I know that, for instance, all of this noise on the right-hand side, AG, AG, ACTG, all this stuff on the right-hand side of this GT, if I know that, for instance, this stuff is my signal, it's hard to recognize. And when I say hard to recognize, maybe people can do that very well. But it's hard to train a machine to do that because a machine, that's really what this subject's all about. We're automating machines to find genetic signatures. It's hard to train a machine to do that because you have to have an idea about what that signature would look like so that you can actually put that recipe of finding it into code. And if you don't have an actual hard and fast rule, if it's all about kind of like looking at things and using a knack and kind of a hunch and blah, 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 well, that's not good enough for machine learning. You have to actually train it with something. And so if every time you're training your software to find these areas, and every piece of training material is different, then it may never learn, your machine learning approach may never learn how to differentiate one boundary from another. Even though the human could do that, though, because maybe they have they have a hunch. They think, well, this is unusual. I don't expect to find this here. But if you're a machine, you have to have all this information there. You have to be kind of told in some ways how to think about that data. So the bioinformatics solution is complicated. It's like, how do we do this? So what you do is in bioinformatics is you have to use basically this probability-based gene prediction method. And that is because you are trying to figure out, like, I don't know exactly what, but I'm trying to think to myself, is it likely that this could happen without being a gene structure piece? Is it likely that I could have a boundary without needing a boundary here? Is it likely that a boundary would be formed randomly? And so a lot of these solutions use probability. One thing that's important, though, is you could use content-based prediction, where that means you're looking at specific types of code in these boundaries. And that's fine, you can do that. But again, alone, it's not very precise. And by that, what I mean is that you need more to go on. How do you tell whether your car is in good health? Well, just by looking at the radiator temperature on their dashboard, that gauge, the radiator temperature, just by looking at that tells you a very small piece of the story. You really have to do other kinds of inspections to understand how healthy your car is. And so, again, when you're looking at a gene, looking at one specific piece is not really usually enough. So what you want to do is you want to combine everything with other types of algorithms. And so that's why, for instance, Monday, when I was showing you like how NCBI, I don't have the slides on my screen now, but how NCBI does their gene discovery. They have all these different stages, all these different tests. We saw that giant tree of all these different ways that information is flowing down this tree, all these different tests that must be done. There may be hundreds of tests that the NCBI does to infer that a gene is a gene. And so you're really using a whole bunch of different approaches to figuring out whether something's a gene. There's no like hard and fast, like look at it, you just look at it and you just know. But rather you're looking at a whole bunch of other things at the same time. But one thing that's actually kind of interesting, though, is the codon usage and these, what they call the CPG islands. And by that, what I'm saying is that you'll have inside your genes, you'll have specific types of syntaxes, or syntaxes, I guess, that are necessary in order for the gene to be a gene. So I've actually published in this area, which is actually quite exciting. So it's fun to see these results again. I haven't seen this stuff in not since, probably not for a couple of years. But this over here, this table is called the codon usage table. It comes from the codon usage database, which is a site in right down here in Japan. But basically what it is that you go to this site. In fact, I can just click on this here just for old time's sake. I'll just have a look and see what's out there. But when you go to this website here, you can type in the name of seemingly any organism you want. Let's go to Homo sapien. So I'll hit H over here, and I'll find human. Well, no, there's human, but Homo sapien. Where are you? Where are you? Something. I'll just click on anything over here. But let's just say on the very, very top of the list here, the human adenovirus type 7D. It sounds very scary, but I'll click on the very first thing. This is an organism. It's a virus. And the virus is actually made up of amino acids. It's a protein, right? And so we're looking at how the amino acids have been written. Like, how does it actually create its amino acids? Another thing I'll tell you about, though, is that if I go to, for instance, codon table wiki, you'll find, for instance, this is my favorite codon table here. This is your codon table. And so, for instance, when you are building protein, remember that you are reading your DNA, well, your RNA in triplets here, or in codons. And so each 3 bases that you find here represents some amino acid or some building block of protein in some way. And so you'll find here, for instance, that leucine can be coded. Leucine is a building block of a protein. It's an amino acid. You can use UUA. I can use UUG, I can use CUU, I can use CUC, CUA, or CUG. All of those will code for the amino acid leucine. In fact, this is where we were talking about the silent mutations, and that maybe traditionally the code would have been CUU, but some kind of mutation happened where now it's CUC. And while we still get leucine in our amino acid chain, it's not the way that the DNA was. And so that is what our silent mutations has come from. So we get that. And so when you look at how different organisms use their DNA, you'll find that how they spell out these common amino acids like leucine or proline or threonine, all of these guys have different ways of being spelled out. Like threonine here has 4, proline has 4, and so on. And so different organisms have a kind of a predilection, if you will, to how they like to spell out their amino acids. For instance, this organism here, this human adenovirus type 7D, this appears to like to use, and these are probabilities here. It seems that in this UUU, what is UUU? UUU is phenylalanine. Phenylalanine has there's 2 different ways of spelling phenylalanine. There's UUU or there's UUC. And so over here, it appears that this organism prefers UUU over UUC when creating proteins where phenylalanine is involved. And so you can look at these different types of organisms and kind of get this idea that, okay, different writers, different styles, different organisms, different styles of building protein. And so that's exactly what you see here. Here we have E. coli and Homo sapien, which have been taken from this website here. And so you're looking at this table here, just the codon table. But you're looking at, for instance, the probabilities or the frequencies, if you will, that these organisms use to write out their amino acids. So interestingly here, I'm looking at the left-hand side where we have the E. coli. In E. coli, for the UUU, which is that phenylalanine, I find that phenylalanine was when you use triple U, that's how E. coli wrote out most of its phenylalines. Not so in Homo sapien. In Homo sapien, Homo sapiens use UUC more in general. There's more frequently, phenylalanine is more frequently used or written out in the C than UUU, which is a contrast. So these two organisms, they both use phenylalanine, but the human uses phenylalanine or spells out phenylalanine differently than the E. coli does. It kind of makes sense if you think about it, though, because, I mean, if we all sat down to write a letter and you spent some time actually looking through the letters to see how we use words, then we would find that I mean, supposing it's a love letter, we're all sitting down to write a love letter. And the way that we describe a feeling that we should all know, love, the way that we describe that feeling is different. We all use different words. This is what they would call a synonymous type of definition here, where we're all talking about the same thing, but how we talk about it is different. And so this synonymous codon usage is different here. So same amino acid, but different codon. Same message of love, different words. It makes sense. Everybody has a different way of describing things, and every gene has a different way of describing its protein makeup. And so you can use that kind of idea to work out a gene. I can say, okay, here I am going through E. coli. I know that E. coli's tend to make up their genes using UUU over UUC for phenylalanine. Therefore, I'm going to suggest that if I find an area where I'm rich in UUU, it could be that that's the phenylalanine that's being used to inject into to create a protein. It's just a small piece of information. But if you look at, for instance, some of these other statistics here, supposing I have all the bases in my codon table. For instance, all these guys here, I have all the probabilities right here for E. coli K12, and so I would use this as a kind of a test. In general, did my E. coli protein, or did my E. coli gene have these kinds of probabilities or these kinds of frequencies for building a protein? And so if that's the case, then perhaps I found my gene area. If that's not the case, then perhaps I haven't, because I'm using this knowledge that's basically what happens. Another thing you can look at, for instance, is the frequency of amino acids. It makes sense that if we're building amino acids, then, for instance, certain types of proteins obviously require amino acids, but you might find that certain types of organisms require proteins that have different constituents or different constitutions of the amino acids. In other words, you might find that there are some organisms that just require a whole bunch of leucine. This L right here that we see on the right-hand side, that's leucine. And so what I'm saying is that you might find that there are some organisms which tend to, where's my table here? Leucine is, where are you, leucine? Here. It's right here. So you might find that there are some organisms that just need a lot of this stuff, a lot of leucine. Looking for leucine in all the wrong places. Just kidding. But what I'm saying is that there are some organisms that just need a lot. And so just by looking at the frequencies of these amino acids, you might think, well, wait a minute. I know that E. coli tends to need a lot of leucine. Maybe, for instance, this area that I'm finding here seems to be doing a lot of coding for leucine, whether it's using these frequencies or not. It seems to be doing a lot of coding for leucine, so therefore maybe this is a gene, because it seems to be asking for a lot of production of leucine. And so you can use that too. And so what I'm saying is that as a probability method here, going back to this idea here, using in combination with other algorithms, one of your algorithms is working with these frequencies for the actual production of the amino acids. Another one is looking at the frequencies of the actual amino acids themselves, like how many amino acids does this organism actually require to build a protein? And so you're using those two pieces of information to infer something. Another thing you can look at though is if you look at the codon usages themselves. Like, for instance, how often are specific types of codons actually used? And you'll find actually that there is a difference. Certain types of organisms do require different amounts of codons or different kinds of amino acids to build their proteins. Arginine, for instance, which is right at the top here, this appears to be one of the more commonly, the highest expected frequency or frequently occurring amino acid to be found. And so you're kind of saying, though, I'm expecting that most organisms are going to be using this because most proteins that I'm looking at for this study tend to have a lot of arginine in them. Now, if I find it does not use as much arginine, or if I find a gene that does not use much arginine, or whatever it may be, then maybe that's not a gene. Maybe that's something else. And so you're using, again, kind of like what you know about those genes. This organism tends to use a lot of arginine. It tends to use it for its genes, for seemingly all of its genes. Therefore, this chunk of code that I found, it could be the gene because I'm noticing that there's a lot of requirements for arginine. So yeah, you're really kind of working again with statistics where you have an expectation of things, and then you have to kind of compare your expectation with what you actually find, and that's actually the hard part in many cases, because what you find and what really is, as you know, if you've studied statistics, will be the same. In fact, it's often not the same at all. And so you really need to have the mindset of a statistician, but also you need to have kind of the idea that you're using previous intelligence, previous learning from what you've seen in bioinformatics to infer how the sequences are created in terms of genes, but also that you're using kind of like boundaries. We're almost running out of time here. Don't worry, I've kept my eye on the clock here, so I'm not going to keep you guys over 10 minutes. I told my friends about that just on the phone just the other day. I said, I kept my class for 10 minutes, and is in medical school in Kansas City. And he said, "Oh, that's nothing. One of my teachers kept us in class for an extra half an hour, because it was once you have all once you have some kind of reaction started, you can't just like say, okay, that's class, and then you just like stop the reaction. It's a chemistry thing. You can't just like stop the reaction and then tell them that to come back the following day when you'll re-establish the experiment. But apparently they're sitting in class and the experiment was just like very, very slow, and it's like half an hour. It's like 10 minutes, then 20 minutes, then half an hour. And then finally, people just began to say, I have to go. I have to go to my next class. I'm sorry. It was kind of funny. But luckily, though, I think that it's more forgiven in places like that. But I do apologize for keeping you guys. Anyway, what I was going to talk about, though, is, and I think that we have some more slides here. I'll tell you what, why don't we'll save these for Friday. I think on Friday, we're going to have another practical. I'll finish these slides here. I have a piece of code for you to look at. And I think we're going to do some more investigations of our own into open reading frames, which I think will be very, very exciting. So bring your appetite for excitement to class on Friday, and then we're going to have our own kind of like investigation of genes here. I think it's going to be exciting. I think it's exciting. I look forward to it. Are there any questions that I can answer before we actually break today? If there's nothing, then class dismissed. I will see everyone on Friday. Take care. Stay warm. Professor. Take care, guys. Stay warm out there. It might still snow, so you have been warned.